So thank you again to everyone for attending. We're very excited in the Science Café to have the speaker. It's actually a day after Rare Diseases Day, so the timing is actually quite perfect. So it is with great pleasure that I introduce Dr. Dang. Dr. Dang is an assistant professor in the Department of Health Sciences at Carleton University. He completed his doctoral studies in statistics at the University of Guelph with a postdoctoral fellowship in bioinformatics at McMaster University. He was previously at the School of Pharmacy and Pharmaceutical Sciences at Birmingham University. He is a statistician and a data scientist who does research in health outcomes and biomarkers, clinical trials in rare diseases, and classification analysis. Dr. Dang's primary interests are related to biomarkers and outcomes in Duchenne and Becker's muscular dystrophy, specifically in understanding the natural history of these diseases through modeling phenotypic, genotypic, and treatment response variability. So I open the floor to you.
Thank you very much, Dr. Casol. Okay, well, thanks everyone for joining to hear me speak today. I'll be talking about understanding rare diseases using statistics and data science. Okay, no disclosures are relevant for this talk. So with rare diseases, the idea is that they're rare individually, but they're quite common together. And the reason being that they're estimated to be about 6,000 to 8,000 worldwide rare diseases. There's no common internationally accepted definition. So the definition in US is different than the one in Canada. But the one in Canada is also the one accepted in EU. And that is if a disease affects less than 1 in 2,000 people, it's considered to be rare. The primary issue with these diseases is that they're not short-lived. They're often chronic and life-threatening. About 80% of them are genetic disorders, and 50% of them affected are children. Now, there's substantial morbidity and mortality worldwide associated with this. As you can imagine, with a rare condition and the amount of research and development that would go into treating that and to helping patients who have that, there is a lot of cost as well associated with this. Now, just as a number to put in, to have this in your heads, combined together, all these rare diseases, the estimates vary, but it's about between 3% to 8% of the population with a rare disease. Okay, so how do we help the folks who have these conditions, these disorders? Well, because of the fact that often the market size is quite small for any pharmaceutical drugs that can help these folks. In these conditions, we have a classification called orphan drug classification. So an orphan drug is a drug for a rare disease. And historically, there have been failures in drug development because the disease was just more complicated than we hypothesized or we speculated to be, or we understood it to be. Clinical trials failed, and so on. And then there was a period of declining investment into orphan drugs. And so over the past few years, both from Health Canada and FDA, to encourage research and development, especially by the pharmaceutical industry, incentives are made available for them to invest into orphan drugs. Now, I should say that it's not just the regulatory agencies like Health Canada and FDA. For rare diseases, parent foundations are extremely impactful in research, education, setting up infrastructure like registries, networks, and so on. And I wanted to mention one little tidbit from CORD, which is a Canadian organization, that there are fewer treatments for rare disorders available in Canada, 40% less, and approval tends to happen 6 years later than in the US and Europe. Interestingly enough, for Duchenne muscular dystrophy, which I'll talk more about in a minute, there's also no drugs approved in Canada, whereas there are about 5 or 6 approved in US by now. Okay, so still keeping this idea of rare diseases, but now I'm going to focus a little bit more on neuromuscular diseases. The area where I work in are muscular dystrophies. These are muscle wasting conditions. And the two disorders where I primarily focus on are Duchenne and Becker muscular dystrophy. Now, these are X-linked disorders, so boys are affected. These are allelic, and I'll talk more about that in a second. They're progressive, so as time goes on, patients who have them do worse and worse. Now, both of these diseases happen because of issues with dystrophin. Dystrophin is a very important protein, and with Duchenne, what happens is the DMD gene, which also happens to be the largest gene in the human genome, has a mutation, which means that the reading frame cannot be completed. And so you end up with no dystrophin production. Whereas with Becker, here, what happens is that, for example, 45 to 47 exons, so there's 79 exons in the DMD gene. If a chunk of this is missing, but the reading frame can still be completed, you end up with a version of dystrophin. It's a partially functional, internally truncated version of dystrophin. So it can still help to an extent, but it's not the same as the dystrophin that unaffected people have. As Dr. Gasol said, yesterday was Rare Diseases Day. I will also mention because of 79 exons in the DMD gene, the International Duchenne Awareness Day is September 7th, just as a little tidbit. Okay, so DMD affects Duchenne muscular dystrophy. DMD affects 1 in 3,600 male births, manifests in early childhood. Symptoms are pretty visible, between 2 to 6 years of age. What happens is because of deficiencies of the dystrophin protein, there's membrane instability, cytoplasmic leakage, and muscle starts getting replaced with fat. And so this overall result of loss of dystrophin in muscle means that there's progressive muscle wasting and early death. There's no cure so far. The standard of care in terms of treatment is high-dose glucocorticoids or corticosteroids. Prednisone is a prime example. We are doing better in terms of a search for better therapies. There's been a recent acceleration, at least for DMD. And there are gene therapies being tested, there are exon skipping therapies being tested, and so on. But they're all supplemented by high-dose glucocorticoids that are taken daily for the best effect. So I have some pictures of patients with Duchenne muscular dystrophy from Defeat Duchenne Canada, MDA and Centera, one of the pharmaceutical companies working to develop drugs. Because this is such a severe disease, it affects your skeletal muscle, affects cardiac, affects pulmonary. We really need a team of healthcare specialists that provide care. And so the economic cost of patients with DMD in Canada has been estimated to be about $345,000 annually per patient. Now, having these patients take daily steroids has helped quite a lot. Over the last 20 years, not just because of that, but that's a huge component of it. The average lifespan of people with DMD has increased from early 20s to early 30s. I want to talk a little bit more about the allelic disorder, which is Becker muscular dystrophy. Now remember, for Becker muscular dystrophy, you still have a version of dystrophin. So you have an internally truncated, partially functional dystrophin. So this ends up being quite, much less severe than Duchenne, but it has a wide range of symptoms. So you will have people, men, because it affects males, you'll have men with Becker muscular dystrophy who have a severe condition and they're in a wheelchair in their 40s, whereas you'll see other folks with Becker who are walking well into their 60s. And the same applies for clinical onset. Whereas with Duchenne, it's visible, in childhood, pretty early. With Becker, some people will not know that they had Becker muscular dystrophy until well into adulthood. So this is how the overall natural history looks like. And by natural history, I mean the progression over time. Behind trying to mitigate the effects of any condition or trying to come up with therapies for any condition, you have to understand the natural history better. And so there are 4 stages for Duchenne, 4 care stages. The first is diagnosis and early ambulatory care stage. This happens in childhood. Then you have late ambulatory phase. This is young adults. So by the time boys with DMD are around 13 to 15 years of age, they've lost the ability to walk and now they're in a wheelchair. And so then the early non-ambulatory phase starts and late non-ambulatory. And the important thing to note is that at each stage, there are different characteristics. Eventually, they will even lose function in their upper body. At different stages, they will need different drugs. They will need different assisted devices, and they will need different specialists to help them. So because this is a muscular disease, the FDA, Health Canada, and so on, they provide guidance that the best time to delay progression in this disease is in this early stage, typically in early ambulatory, but also possible in late ambulatory. And this is because, as I mentioned, muscle is being replaced by fat. So you want to have some muscle left over that you can actually delay progression before it's too late. So this is also the period of interest to clinical trials of new investigative drugs. Okay, so I want to talk a little bit about what kinds of data we get. Remember, these are rare diseases, and a typical clinical trial in Duchenne muscular dystrophy will be 100 to 200 participants, no more. Okay, and this will take place in like 15 countries around the world. So you're getting really quite few people. But what you do collect is a lot of data. So I want to talk a little bit about that. So we can get clinical outcomes. You can get direct outcomes that measure strength, like myometry, or you can get data on clinical outcomes that measure function. So for example, how long does it take to get up from the floor? The reason a data point like this is important is because boys with Duchenne fall. And when they fall, it's important for the families, it's important for the parents to see how long for them to be able to get up. And so this has been used as a primary outcome in clinical trials. But there are other outcomes available as well. You know, some will test function in terms of proximal muscles, others will test acceleration, how quickly you can do something, others will test stamina still. Because they're taking high-dose glucocorticoids daily, their bones also are more at a risk of having fractures, becoming brittle, and so on. So we also collect specialized data with X-rays and MRI and so on. High-dose glucocorticoids also gives other types of issues, growth stunting, behavioral issues, Cushing syndrome, increased weight, and so on. It also leads to adrenal insufficiency, secondary adrenal insufficiency, where your body makes less and less of cortisol endogenously. And so all of these things are captured. Well, this is all well and good, but often what matters is the quality of life. And so we also collect data from patients directly, rather than as a clinical outcome. We collect data on patient-reported outcomes on quality of life. So for example, are you able to shampoo your hair? Are you able to get up off the floor from lying on your back without help? Not just the time required to do so, but are you able to do so? And of course, because it's a genetic disease and we want to know we want to know more about the pathophysiology and what's happening in the body, we collect a lot of biomarker data as well. So this can, takes the form of mRNA, microRNAs, proteins, SNPs, also depends on which particular mutation was leading to this condition. We can collect biopsies data, but of course it's more invasive, or we can just take a blood draw and quantify protein levels from there. Recently, we've also started talking and getting data on digital markers. So this is like using a Fitbit or an Apple Watch to get free living activity data. And all of these are affected by the drugs these patients are on. So what kinds of challenges do we still have? Well, for patients, we want to be able to reduce the burden of disease. And because it's a rare disease and because clinical trials tend to be small, we want to make sure that the burden of being involved in a clinical trial or an observational study is also less. So we want to be able to do that. We also want, of course, primarily we want to help increase the length and quality of their life. For clinicians providing care, we need better outcomes that capture what's happening in the body. The most sensitive outcome that we've seen is, the level of fibrosis, the level of how fast muscle is being replaced by fat, but it's difficult to get at that quantity easily. We also want to be able to provide to clinicians better prognostic milestones. If someone is unable to do a time to stand from supine test, what does that mean? Should they be changing the treatment based off of that. And also to be able to explain why is it that some patients have a ton of side effects from glucocorticoids while others don't, and other drugs as well. For the pharmaceutical industry, the problem has been that lots of trials have failed because of low statistical power. And that typically happens because of a low sample size. And that happens because it's a rare disease and you can't enroll a lot of people. But it's primarily complicated by the fact that there's incredible heterogeneity seen in the natural history. And I'll talk more about that in a second. So it's really the intersection of it being a rare condition as well as there being lots of variability in outcomes seen. Just as a, just to provide some more background, the kinds of data that I work with and the way we are trying to do some contributions is through multiple clinical trials, ancillary studies from those clinical trials or observational studies, cohort studies, cross-sectional studies. We really try and use the best study design for the question of interest. Okay, so some key questions are, well, can we distinguish between clinical severity from treatment non-response? Some of the clinical trials that failed, it seemed like if we had a larger sample size, they could have succeeded. But it's unclear because you can have a patient who is so severe that treatment cannot help them at all. So how do we distinguish that? Can we use biomarkers more and more often? And this is certainly a direction that the field is going in. Can we de-risk clinical trials via biomarkers? Can we predict those who will have good treatment response really quickly to try and cut down the amount of time it takes to run a clinical trial, or if you have to start from scratch, do everything over and over again? Can we reduce patient burden via biomarker signatures? Is it required that these patients, who are dealing with this condition have to go to clinical sites every few months, or can we just send a phlebotomist home or a nurse home, get a blood draw from them and just be able to understand what's going on in their body using that? And can we enhance clinical trials enrollment or monitoring via biomarker signatures? All of this has to do with how do we conduct well-powered analysis? How do we keep our trials efficient and so on? So here's an example of a clinical outcome. So this is the time to rise from supine position. How long? We will use the stopwatch. How long for this boy to rise? Here's another outcome. This boy is walking to this orange cone and will walk back, and they will do so for 6 minutes, and we calculate how far did they walk. So it's a 6-minute walk distance score. And of course, we can only use them while they retain ambulation, while they can still walk. After they've lost the ability to walk, we have to rely on other outcomes. So there are some other outcomes as well. Time to climb 4 stairs, time to run, walk 10 meters. This captures, this is capturing acceleration. This is still capturing proximal muscles and so on. And some of these tests are easier than others. And so when you look at lots of observational data, you'll notice that the time to stand from supine test is the first to go. So it'll start showing up as missing data, but really it's because of disease progression, these boys aren't able to do it. And so the ability to be able to model that properly is also very important. So here is some very messy data from a prospective study, a retrospective study. This is 6-minute walk distance. So remember, this is how far can these boys walk in 6 minutes. This is 0, this is 800. So the further they can walk, the milder they are. This is a North Star ambulatory assessment. There's a ceiling effect at 34. This is the maximum it can be. Again, 0 is very severe, 34 is very mild. I want to point out that, there are 10 to 15-year-olds who are doing really, really quite well here, being able to walk 400 meters. And then you have all these other boys as well who are extremely severe. And there were some boys who had lost the ability to walk at age of 8. Same thing happens with North Star ambulatory assessment. Not quite at 0, but their scores are declining. And each of these lines is a patient over time, so it's longitudinal data. So to understand this a little bit better, because the worry is that if you end up with too many boys who look like this in a clinical trial, well, maybe they were too severe that the drug could not have helped them. And maybe the drug did work, but we saw that the trial failed because we had too many of boys who were too severe. So to try and understand that better, what we did was we combined data from real world prospective cohort study with all sorts of treatment regimens, boys not taking daily steroids, they're taking weekend steroids, taking intermittent steroids and so on, switching between different drugs, along with clinical trial data. We looked at, instead of looking at the time directly, we also looked at velocities of how quickly can they stand up, how quickly can they walk up 4 steps, how quickly can they walk or run 10 meters. And we also modeled properly missing data. Did it mean inability to do something? Was it because of an injury? Was it because of lack of time? Was it a bad day that this boy was having, and so on? So what we conducted was a longitudinal latent class analysis of these early age outcomes, and we analyzed multiple outcomes simultaneously together. And the importance of doing it this way is that all of these clinical outcomes are correlated, but they're also capturing slightly different things. So saying that one boy, is doing really well on this particular outcome is not enough on its own. And saying another boy is doing really poorly because they have a poor score on this one particular outcome is not enough on its own. And so what we were trying to do was cluster similarly progressing boys over time, and then see if there are any interactions with pharmacological therapeutics. So it's extremely messy data. So we have climb velocity, we have stand velocity, and we have run walk 10 meter velocity. So as I said, each of these boys is a single, each of these lines is a single boy. So you can see there's lots of intra-subject variation, lots of moving up and down. It's messy. Some boys seem to decline very quickly. Other boys are doing really quite well until much later. And so when we fed this through this clustering model, what we noticed was that there seemed to be 3 different classes of boys, and severity is on a continuum. So there's some sort of, we're doing this categorization, but we're doing this to understand the natural history better. In the top row, we have stand velocity. In the middle row, we have climb velocity. In the bottom row, we have run walk velocity. And this first group, approximately 10% of the boys, are the fastest progressor groups. Notice that their scores, when high is good, just go down to 0 very, very quickly, all before the age of 10. On the other hand, the slowest progressors are doing quite well until much later, right? They're not so many 0 velocities in the slowest progression group. The moderate progression group we found to be of quite a lot of interest. The expected trajectories that are here, the blue is substantial early life steroid exposure. And the idea here is if you're exposed to steroids early that are dealing with the inflammation in your body and some other things that they do, well, you're able to retain that performance bump for longer. And that's what we see here as well, is those who were exposed to steroids early tended to do better, tended to have better outcomes. And the difference between substantial early life exposure and being steroid naive, the difference was the largest in the moderate progressive groups. And the way we hypothesize about this, is that if you're too severe, drugs are not able to help you a ton. If you are quite mild in comparison, drugs are still able to help you, but not as much because there's still a ceiling on it. So it's really the moderate progressors who are seeming to fall into this Goldilocks zone, right? It's just right. And so then the question becomes, well, can we identify those who will benefit the most from treatment early? Because if we can do that, we can try and enroll them in clinical trials for any drugs that can help these boys. And if we had to do that, we would want to make sure that we don't have a ton of fastest progressors in our clinical trial. And yet we want data on everyone. So what we can use is, if we had a validated prognostic model at an early age to be able to understand, how this boy will react over time, how this boy will do over time, we could use partial enrichment strategies or planned subgroup analysis, enroll everyone, but focus on our primary endpoints on a specified subset of groups, specified subset of participants. Okay, so that was clinical outcomes. I want to talk a little bit about digital markers, actigraphy and accelerometry. So I'll say promise of this type of data because it's unclear how much it's helpful at this particular time point. First of all, what do these measurements reflect at the patient level? So this is just like having data from Fitbit, but this is an actigraph, which is an FDA-approved device. What we're getting is free living data. With a clinical outcome, we get peak performance. We tell those boys to do it as quickly as they can. With this is free living data, and the goal is, well, if a boy is milder or they're doing better because of treatment, this should be reflected in their free living patterns. And we get data on activity and sleep as well from it. But there's all sorts of things, because it's truly free living rather than structured, there's all sorts of things that affect what kind of data we get. It's also been unclear, where do we place this data? Where do we place this device to collect this data? Makes sense to intuitively place it on the wrist, but there's been better performance seen with waist and ankle, actually. There's also the issue about how do you collect data that's in accelerometer units to be able to convert it to activity counts. And so there's some unclear processing algorithms. There's also poor adherence. In an ancillary study that we were part of, boys kept losing the device, so they didn't want to wear it and so on. Next up is biomarkers. I want to mention that, so this is, again, the time to stand outcome that I was talking about. And this has been converted into velocity, as I mentioned. So higher is better. This is a clinical trial that I was a part of running. And what we saw was this. In the gray, we have boys who were randomized to a placebo group. The data is on change. Each time point, each point corresponds to a particular time point, week 6, week 12, and week 24. Notice that the boys in the placebo group, their mean change, of course, at baseline, we're comparing to baseline, so it's 0, it's declining over time. Whereas boys who were randomized to vamorolone 6 milligrams per kilograms per day, which was only 28 boys, they are doing better over time. Well, that's great. And the fact that we were able to have a clinical trial actually show clinical efficacy was a big win for DMD, really. But it also takes time. It takes time to gather all these data, and there have been lots of drugs that didn't succeed. And so what this drugs program had done was quite innovative. They de-risked with established disease-specific markers that reflect the mechanism of action of this drug. So vamorolone, like prednisone or deflazacort, which are the standard of care, is an anti-inflammatory, but it does certain things in a different way. But what it did in just 2 weeks of its phase 2A clinical trial was it noticed that these inflammatory markers, CD23 and MDC, which are quite high in a Duchenne population, also decline in a dose-specific way. So higher dose, there's more decline over time. And again, the idea being that you connect the anti-inflammatory bit to the data that we see, you can see that quickly enough. So you're de-risking the program by connecting your biomarker levels to what hopefully you will eventually see in the clinical outcomes as well. And you can connect this with pharmacokinetic levels. Did these boys have a good exposure to vamorolone? And how did this compare with boys who were taking 3 times the dose and so on? So you can look at Cmax levels, you can look at area under the plasma concentration time curve levels as well. I also want to talk about sort of genetic modifiers. Of course, as I said, this is a genetic disease. But at the same time, not everything is defined by the fact that dystrophin is not being produced, because there are multiple mutations that lead to DMD. And what we've noticed is that there are some mutations that are much milder than others. And one of the reasons that has been identified for that are these common polymorphisms in other non-DMD genes that have generally been found to involve fibrosis and inflammation cascades. Phenotypically, what does that mean? That means that, you know, boys who have a particular SNP, they might end up with 2 years extended ability to walk, or 2 years less ability to walk, and so on. But this type of data is often complicated by low allele frequency seen in one particular country. So we found, you know, we've seen in the field, some SNPs end up being statistically and clinically significant based on population from one set of countries and not relevant in other countries. There's also, of course, there are interactions with social determinants that are not often modeled. We're conducting a study right now, and we got data from a SNP chip, which had 1.9 million markers, but of course, it's a rare disease. And so the worry is that you use all of this data, try and do a discovery, and you have to correct for multiple testing considerations. You end up with underpowered analysis and you get nothing to move forward on. So instead of doing a full discovery, what the approach that we have taken is we actually do a targeted study. So not a full genome-wide association study, but instead a targeted study on some SNPs that are of interest. I'd mentioned patient-reported outcomes before. I want to talk a little bit more about that. But this time switching gears a little bit to Becker muscular dystrophy. So with patient-reported outcomes, rather than something you measure, you know, based on motor function or something like that in the clinic, that's clinical outcomes. Here we ask the patients to answer a question. And what we're trying to get at is quality of life. And so we used a questionnaire called NeuroQOL, which is quality of life in neurological disorders. And it captures data on these different subdomains. One of the subdomains is this umbrella is the mental social subdomains, and it captures data on anxiety, depression, as well as fatigue. And I'll show you some data on this. This is unpublished, it's an ongoing study. And I'll show you that a higher score means that they're doing worse. It also captures quality of life data on their motor capabilities. And so you capture data on upper extremities as well as lower extremities separately. And in this case, lower score is actually worse, higher score is better. So two sort of umbrella subdomains, mental, social, as well as motor. So this study, we had 83 males who had up to 3 years of follow-up with annual assessments. Data was collected from 2013 to 2018. And I'll show you two of the subdomains. I've split it at age 18. So minors on the left hand and adults on the right hand. And notice that this is anxiety. So higher, the worse. So this participant is, was based on their reported data, was highly anxious. Okay, at this time when they fill out the questionnaire. They're also supposed to have up to 3 years of follow-up, and this participant only contributed 1 data point. So they might have refused to answer questions. Other times they might have entered the study too late, and so on. So what you get is, again, this sort of, continuation of you're getting a lot of messy data. There's lots of attrition, there's lots of missing data. On the right-hand side, you have anxiety scores for adults. And notice how much it varies, right? So the lowest it can be is 8, the highest it can be is 40. And you have men whose scores are varying within 1 year from 25 all the way down to 8. And so it's really capturing quality of life. But what's of interest is that during this up to 3 years of follow-up, it's overall flat. right? There's change in it, but if we had to fit a longitudinal model through it, there might be a slightly declining slope, but overall there's a little change happening. It's just a lot of variation. Compare that to lower extremity scores. With minors, and in Becker, most minors are doing perfectly well. The progression really is in adulthood. With minors, there's some variation, but overall, they're actually doing quite well. And remember, for lower extremity score, higher is better. So they're all clustered around here. They're doing quite well. But for adults, there's lower extremity score. You can see this sort of trend of decline over time. And so this is an alternative to capturing data via clinical outcomes. So this is one way we could cut down on the burden on patients, is by, if we don't need to get them to come into a clinic, we don't ask them to, we just let them answer a question from home. We also looked at correlations between these subdomains and correlations with motor outcomes. And what we found was that the lower extremity score really does correlate somewhat well with clinical motor outcomes, but correlations between the social mental subdomains as well as the extremity subdomains was not quite high. So it suggests to us that it's not just how much they're declining in terms of their motor abilities that's leading to anxiety, depression, and fatigue. There are other aspects at play as well. Okay, so that's a lot of different types of data. Now, we want to be able to combine all of that so we can do multivariable modeling. So here I have just a little figure. So this is patient A, visit 1, patient A, visit 2, and so on. And so you can have clinical outcome data, some demographic data, some genetic data, and so on. And you can have lots and lots of biomarker data. Thing is, this is very, very wide data. You have lots and lots of dimensions, but you have very few patients. So the largest prospective cohort study that was done in Duchenne was 440 participants. And this was done in many countries around the world. So it's been difficult to apply advanced machine learning tools here. There are issues of validation. It's still black box. There's too much heterogeneity combined that with small sample size leads to issues. So what we do instead is we use biologically motivated, explainable statistical learning models. So I'll give one example of what we have done before. We had data from this aptamer-based instrument. So we had somologic data, and we had data on 1,300 biomarkers on just 39 participants. So trying to model this with a regular regression model, a regular multivariable regression model, just doesn't work. And so what we did was we identified different clusters of proteins that were working very, very similarly in terms of their expression patterns, not in terms of quantity alone, but in terms of their patterns. And once we identified those clusters of proteins, we also identified within those clusters which were the most representative proteins and the most well-connected proteins. And we only carried those proteins forward into multivariable regression modeling. I won't spend too much time on this. The details are in the paper listed below, but basically that involved clustering these proteins, then building using statistical learning models like ElasticNet and Lasso, these much narrower, much smaller models. So we went down from 1,300 proteins to 17 proteins to be able to explain time to stand associated severity, or 6-minute walk distance associated severity. And then once we had that model, we did outcome validations. We did comparisons of the muscle biopsy gene expression versus histological severity. We tried to build some biological models to explain why our model worked. Okay, so there's been a lot of progress in Duchenne, not as much in Becker. There's still a lot to be done in Duchenne, of course, but we are starting to conduct more studies in Becker. I'm involved in one clinical trial in Becker right now as well. And what we're trying to do is transfer some of these lessons from rare diseases in general, but also Duchenne to Becker. So we're trying to think about who would be a rapid progressor versus who will have a stable trajectory. What age range should we be running these trials in? I mean, we don't even know Yet, is it better to be running a clinical trial in Becker in 30 to 40-year-olds, 30 to 50-year-olds, or later than that, or younger than that? It's unclear because Becker has even more variability in their phenotype than Duchenne does. Can we simultaneously de-risk via biomarkers? How much change is really possible in a short clinical trial? And how do you run a study? How do you do a sample size calculation if there's never been a successful study run in this patient population before? So all those things we're taking lessons from one muscular dystrophy, Duchenne, transferring it to Becker. Okay, some closing comments. It's really important to have good experimental design and use statistically powerful data science models. It's really important to have head-to-head studies, especially in a rare disease. You do not have large sample sizes to save your conclusions, so to speak. Head-to-head studies are super helpful. Crossover studies are super helpful. It's important to have standardized outcomes and how do you measure them and how do you interpret them. So if you're doing bone x-rays, you should use central reading. If you are using continuous data, don't dichotomize, use the continuous data. If you have lots of missing data or sensor data, use models appropriate to data because you have, the sample size is precious. You have too few data points in the first place. And then of course, there's no one size fits all approach. What worked for one clinical trial in a particular age range will not necessarily work for another in another age range. And of course, we also want to try and reduce the burden on patients. So don't ask them to do these outcomes that are unreliable, because if outcomes are unreliable, you can't tell whether something changed or was it just an unreliable outcome in the first place. We're also thinking about how to increase enrollment. We're talking about increasing compassionate use possibilities where clinical trial companies or pharmaceutical companies will allow the patients who were part of their studies to get drug for free after or until the drug is approved. We're talking and thinking about how do we share the clinical trial data back with patients, because this is not a thing that has been done before in a systematic way. And some patients really want to see that data for themselves. We're talking about how to build larger data sets so that we can do meta-analysis. There are challenges in this. The recent, so GDPR has given us some challenges in terms of pooling data. Some of our collaborators are now saying they would like to reconsent patients to be able to send data to Canada and so on. And then anytime we pool data together, always keep in mind, there's ascertainment bias. Who was seen in one particular population in one particular study versus another? How does that affect? How do we combine these data sets together? There's also possibility of biomarker-based, cheaper remote studies and using quality of life questionnaires and so on. I'll leave it with this, that even though we end up with very messy natural history data from these conditions with complex pathophysiology, this can still be extremely informative. Okay, so I have some references here. I just want to thank some frequent collaborators and trainees who were there from Binghamton University, University of Pittsburgh Medical Center, CHIO, Children's National, and so on. The funders, Foundation to Eradicate Duchenne, NIH, and DoD, and of course, huge thanks to the patients with muscular dystrophies and their families for participating in these studies, as well as all the clinical coordinators, evaluators, the Synergy Network, and Trends. Thank you.
Great talk. Thank you very much. So the floor is now open for questions. So if you have any questions, please type them into the Q&A box and I will read them to Dr. Dang. Maybe while we're waiting for that, I can start. So it was quite striking, the difference in the level of approvals in Canada for drugs to treat rare diseases. Could you tell us a little bit more about why that might be happening or why it takes so much longer in Canada to get approved?
I don't know much about that, unfortunately. I do know that many patient foundations, many parent foundations have been calling for a national rare disease strategy. Looks like it just seems to me that it often ends up being deprioritized in lieu of other things. And so I wish I knew more about that, but still learning about that.
Yeah, no, that's
Question,
right.
though.
Okay, so we have our first question in the question and answer box. So have you heard of any clinical trials for Becker MD that showed success using DMDM modulators like, I'm going to butcher it a little bit, Trifecta or cystic fibrosis? Sorry. So are there successes using DMD modulators in Becker MD? Any clinical trials that you're aware of?
I'm only aware of 2 clinical trials that are being enrolled in Becker muscular dystrophy right now. One is the trial that I'm conducting, which is using vamorolone, and the other is by this other company called Edgewise Therapeutics. And no, I'm unaware of Trifecta being used for Becker muscular dystrophy or DMD modulators like that.
Okay, great. Any other questions? I have one more, if anyone, just to give everyone a little bit more time. With regards to then sort of the clinical trials and these rare diseases and the fact that, you know, the N for these studies is always going to be low. So is there some need for the system to change the parameters or the definitions of successes for clinical trials in these types of contexts? Do you think there'd be any benefit of that? Or do you think there's nothing that would, something like that would never happen because it's such a defined process for the approval of drugs and clinical trials?
So, no, that's a great question. Some of the drugs that have been approved in the US recently have not been approved based on clinical efficacy. It wasn't based on time to stand or time to run, walk, or anything like that. They were based on the fact that these were specific classes of drugs called exon skipping drugs. And what they were trying to do was, using a drug, convert the Duchenne phenotype to a Becker phenotype. So this drug is designed to skip over exons so that the reading frame can still be completed and you can have some partial dystrophin. Now, what we've seen so far is it's quite low, but it is successful. But there are drugs that can, that induce up to 5% of this internally truncated dystrophin. And so they have been approved based on this surrogate of the fact that there used to be no dystrophin before in these men, and now there is. So just based on that. And then we're continuing to collect data over a longer time to see if it can connect it to clinical efficacy. But it does seem like that should help as well. So that's been one alternative way, is that instead of waiting for clinical outcomes data, we do it based off of biomarkers.
Okay, great. So the next question is, could you explain again the concept of de-risking?
Yeah. So the idea there was that with a early phase 2 clinical trial, right? So phase 1 would be first time in healthy patients. With an early phase 2 clinical trial, the idea is this is really the first time patients, people with a disease are being exposed to this drug. And so if our definition of success was follow up over 6 months or a year before we evaluate whether this is working or not, that can take a lot of time. So the idea of de-risking the program was, well, if we are designing a anti-inflammatory, well, the inflammatory biomarkers should reduce in short time, very, very quickly, much quicker than you would see a change in clinical outcomes.
Okay, great. So the next question is a bit trivial, but what's the ratio of researchers to patients? I assume that patients get much more personal attention from researchers than ones with common diseases.
Oh, I
guess,
don't know
yeah.
the ratio. I would say that there are, it's great that there's lots of attention towards these rare diseases, but the fact that we have fewer patients and they're spread over a very wide geographical area also means that there's not a lot of specialists and experts. And so often these patients have to travel a fair distance to be able to go see an expert.
Right. Yeah. No, takes a lot to get these things going. So next question. Did the heterogeneity of symptoms make it difficult to define or discover biomarkers for these diseases?
Yeah, that's a very good question. So when we define biomarkers, or when we do discoveries for biomarkers, we basically start by which care stage are we interested in? Are we interested in a biomarker that will help us diagnose it early on? Are we interested in a biomarker that will help us think about how a particular boy is doing? So that would be a monitoring biomarker at any given time. Are we interested in prognosis? Are we interested in an early stage where they're still walking, or are we interested in the stage after they have already moved into a wheelchair? So all of those things are nuances and really cut down on the sample size even more, unfortunately, because the biomarkers end up reflecting what's going on in your body most of the time. And so we have to deal with that as well. Good question.
Well, thank you everyone for your attendance, and thank you, Dr. Dang, for this excellent presentation. And yeah,
thank you very much.
Hope everyone has a great day.